Hello friends, today I am back with a new interesting topic. I am Kanchana on my YouTube channel. You can see that in every family there are same pattern of face, nose, eyes, habits including diseases because the blueprint present in our gene that inherit generation after generation, that all minute details present in our genes. So today's topic is nucleic acid and DNA, that is most important. So if you are not subscribed my channel yet, then please subscribe. Thank you.
Nucleic acid, another biomolecule, contains nitrogenous base, pentose sugar, and phosphoric acid.
If you see the structure at a glance, it becomes headache for you. But I'm sure after watching this video, your brain will love this topic. Now let's start.
Nitrogen bases are of two types. One is purine, another is pyrimidine. Purines are heterocyclic hydrophobic double benzene ring structures, and pyrimidines are single ring hydrophobic heterocyclic structures. One is like that and another is like that. Now, purines are of two types. One is adenine and second one is guanine. Simultaneously, pyrimidine are of also two types. One is thymine and another is cytosine. There is another pyrimidine present in RNA, that is uracil, but we will talk about RNA on the next video. Now, if we add purines or pyrimidines with the deoxyribose sugar, then it will be called deoxyribonucleoside. And if we add deoxyribonucleoside with the ions of phosphoric acid, that is a negatively charged compound, then it will be called deoxyribonucleotide. Now, the full form of DNA is deoxyribonucleic acid. It means deoxyribonucleic acid is an acid that have a sugar of deoxyribose type. The structure is like that. The number 1 carbon is that, the 2 number is that, 3, 4 and 5. In case of 2 number carbon, there is not any oxygen. The 2 of R are only hydrogen. It means deoxy, the lack of oxygen on the 2' carbon, since the name is deoxyribose sugar and the name DNA. Because of the phosphoric acid charge, the DNA is also negatively charged compound. Now the structure of adenine, guanine, thymine and cytosine are like that. First, adenine. It is a double heterocyclic structure like that. C, N, C, N, C, C, N, C, N. In this portion, the N and C position will alternately placed, and only in the middle position it will be only C and C. That we know, N has a valency of 3 and C has a valency of 4. Then we will adjust that according, and we will number those atoms like 1, 2, 3, 4, 5, 6, 7, 8 and 9. Next, on this position there will be a main group, that is amine, NH2. Then a bond in that position and a bond in that position for a sugar of the deoxyribose. Now we will place a thymine for bonding with adenine. This is a heterocyclic structure of a single benzene ring. CN. The bonding pattern will be similar, and we will add a keto group on that position. On that position also, we will place here the sugar again, and we will put a thymine here. It is a special character, we have to memorize it. Then we will place some hydrogen bonds over there for the bonding pattern. Adenine and thymine has 2 hydrogen bonds between them. The electronegative atom and the electropositive atom will create a hydrogen bond here and here. Next, we'll go for cytosine and guanine again. CN. We'll put a amine again here. Then adjust the bonds. Now we will put a guanine for bonding with cytosine. It is again like adenine. CN. We'll put a keto here, then the hydrogen there, a amine is there. Guanine also have amine as well as keto. Now C and G have 3 hydrogen bonds similarly with those groups 1, 2 and 3. So the CG bond is more stable than A and T. Now we have to put the deoxyribose sugar with these nitrogens. One, 2, 3, 4, 5 CH2, and if we put OH there, then we have to add the phosphate group with that CH2OH. So I am not writing the OH here, I will add the phosphoric acid there. Now I am adding the phosphate with the another sugar and then a phosphate also. So here you can see that the 3' the 5 prime. It means the 3 number carbons OH is ready for another reaction, and the 5 carbon here of phosphorus is also in a reactive position. So in this case, this 5 denotes the 5 prime strand of the DNA, and this 3 number carbons OH denotes the 3 prime of another strand of the DNA. And this manner is fixed. This is the phosphate sugar backbone that is present outside, and these bases are present inside. Simultaneously, I am adding these strands on this position. You can see there the 5 prime and the 3 prime group as the opposite direction. So in this case, the two strands are opposite. One is 5 prime to 3 prime and another is 3 prime to 5 prime. Now the bonds between them. So the bonds between the base and the sugar, this bond is called glycosidic bond. This bond is also called a glycosidic bond, this bond a glycosidic bond, and this bond a glycosidic bond. And this structure made by phosphoric acid, so the bond is phosphodiester bond. There is also phosphodiester bond. This bonding pattern is already told by scientist Chargaff. He said that adenine has 2 hydrogen bonds with thymine and guanine has 3 hydrogen bond with cytosine. But this equals to doesn't mean about the bonds. It means he told that the amount of adenine is equal to amount of thymine, and the amount of guanine is also equal to amount of cytosine. It means the amount of A plus G by the amount of T plus C is always equal to 1. It means A plus G is equal to T plus C. The both two are purines and those two are pyrimidine. According to Chargaff's rule, it is clear that the amount of purines is equal to amount of pyrimidines. If we assemble the whole thoughts in one page, then we have to consider the Watson-Crick proposal that they told that DNA is present in always a double helix manner. You can see here that 2 rows are present and both strands are twisted like a helical manner, and the characteristics are they are opposite double helix. Opposite means if one strand is 3 to 5 direction, the other will be 3 to 5 direction in opposite manner, and this cause the nature anti-parallel. The 2 strands should be always in anti-parallel manner for their stability, bond forming and other thermodynamic consequences. They are intertwisted around each other. They are already twisted. If the structure is like a ladder and there is a bond between them, then if we twist the 2 strands, the structure will be like this. The next character is right-handed helix. Right-handed means the helical turn will be on the clockwise manner. If it will on anti-clockwise, it will be left-handed. But the biological DNA is always right-handed helix. Next, the two strands held together by hydrogen bonds. We know that these bonds between them are hydrogen bonds. We are talking about these hydrogen bonds that present in the helix. Next, the bases are stacked on core sugar phosphate backbone at the outside. We can see here that in the helix, the ladder is made up of sugar and phosphate backbone and the bases are present inside. If there is a adenine there, and the complementary thymine should be there, and in between them two hydrogen bond will form. Simultaneously, if cytosine is present there, guanosine should be present there, and three hydrogen bonds will make them structure stable according to the Chargaff's rule. Next, the other physical characters. The diameter of the double helix structure is 20 angstrom. Next, the turn per base, there is present 10 base pairs in the structure in the turn, and the length is 34 angstrom. So the simple mathematics said that one base is equal to 3.4 angstrom because 10 base pairs are present over there. And another two angles are situated there. One is greater than 180, that is called major groove, and one is less than 180, that is called minor groove. And these characters are told by Rosalind Franklin and Wilkins from the London King's College. They told it by X-ray crystallography is taken by them and they measure the structures. The two strands should be complementary to each other as always. DNA is so important because
it carries every minute details of our genome and responsible for all metabolic activity. So this is the gist of DNA. I will be back with another topic. Till then,